Hey, so this is going to be your second lesson to your Punnett Squares, and continue from the last video about relating meiosis to Punnett Squares. You now know that if we were to start off with a cell with these chromosomes, I'm just going to quickly draw some chromosomes here. Here's my centromere in the middle. So these are your two different chromosomes, and if one of them accounts for your blonde hair color, and this one accounts for your brown hair color, we know that during meiosis, these ones are going to duplicate and cross over. That means that they're going to duplicate first, and I'm going to change colors so then you don't get confused by this. So they're going to duplicate. I should have changed colors earlier. So let's make this purple one. So then after that, then these ones are going to sort of cross over, and then the next stage of my meiosis is that I'm going to have a crossed over chromosome so that they created a diversity. So here's one, and then one of them is going to look something like this, and then I'm going to have my purple. Let's see right here. So then I'm going to have this purple that's been crossed over, and then I also have these ones, and then one full one, and I'm going to have my yellow one that's been crossed over. So this is sort of my homologous chromosomes that has been duplicated. So the next step is that these ones are going to segregate. So the first law of Mendelian genetics of this whole thing is segregation. That means that all of these chromosomes will go into its separate sort of sex cell or haploid cells. And what happens, you know that at the end of meiosis, you're going to have all four haploid cells that's been generated. So let's see if I can just do this very quickly. So one of them is going to be, oh, that doesn't work. I'll just have to draw this out. So what happens is that one of them is going to have the first one. So this is my first chromosome right here. And then I'm going to have another one that looks something like this. That's been crossed over with the purple right here. I'm just going to draw this. Oh, never mind. And then the purple one, like here, purple. I just need to fill in the yellow one. So you can see, hopefully by now you understand what's going on, that all of these ones have crossed over. The traits have sort of mixed in with the entire genetic pool. So all four of these are your sex cells, your sperm and egg cells. So how does this relate to, how was this first discovered? Well, first thing that was discovered was by an Austrian monk by the name of Gregor Mendel. And what he did was that he did a lot of pea genetics, whereas he looked at the pea plants and bred them and looked at and counted the number of plants that he got as a result. And it was a very meticulous task because he had to count lots of different and collect tons of data to form his conclusion. What he found was that one of his experiments that he bred these purple pea plants with another one, but what he noticed was that he didn't just get purple plants, but he also got a white flower as a result. So what does that mean? How come he only got purple and white, and how come it was a perfect sort of 3-to-1 ratio? So what he noticed was that, and concluded, is that perhaps in Mendelian genetics is that your genes are either one or the other and not a blended version. So first law is that you're going to have, first law of genetics is that you're going to have the law of dominance. This is where the dominant trait, such as this purple flower, is going to dominate and be expressed over the white one. So the purple flower is what they call dominant. And what we use is going to be at the uppercase letter. So that's your uppercase B. The white flower is what they call recessive. So recessive is going to be used and depicted by this lowercase b. So what that means is that in order for a dominant trait to occur, actually that's the easier one, because the dominant trait will just be expressed anytime you have an uppercase genotype. So anytime you have the uppercase letter, that dominant will overshadow and really be dominant over the recessive one. So when you have two uppercase B's, let me just kind of change the color here. When you have two uppercase B, this is what they call, oh, I thought it changed colors. When you have two uppercase B, this is where you have what you call the homozygous dominant. So that means you have both of the copies. So this is uppercase and uppercase. If you have one or and the other, this is what you call a heterozygous. So this is a zygous trait. So you have one and the other one. So one of both traits, or one of both alleles actually. So uppercase B, lowercase b. And in order for this is going to be your dominant one that will show. However, for the recessive one, when you have two lowercase b's, this is what they call your homozygous. Same thing, homozygous means you have two of the same letter, but now you have homozygous recessive. This is where you have two lowercase letters, and this is the only way that the recessive trait can be shown, is that if you have both recessive genes. So that's how you, these are some of the words, key vocabulary that you need to know from Mendelian genetics. Another key term that you need to know, let's see if I can just move this down a little bit, is that what they call alleles. And I haven't really touched too much on that yet, but alleles, let me change to green, I'll just write here. Allele is, what that means is that it's just a different version of that gene, alternative version. So if we're looking at just hair color, brown is an allele, blonde is an allele. Both of them still account for hair color. If we were to look at eye color, blue is an allele and green is an allele of that trait of eye color trait. So when you hear the word allele, that means it's just a version of that trait. Okay, so this is what sort of the basics of the Mendelian genetics and setting up the Punnett square. So let's try a question here, is that, let's look at this one. In dogs, a dominant gene W produces wire-haired texture. So we know that the dominant gene is going to have the uppercase W, and that means that it's going to produce a wire-haired dog. So those are the fuzzy kind of dog that you see. Those are usually terriers and kind of cute little dogs. Right here, let's just draw two eyes. Wire hair. The recessive allele, that means the other version, is going to be the lowercase w. I always underline my lowercase just so I know, because w's can get very confusing. Produces a smooth hair dog. So here's my smooth hair dog. There's no fuzziness, it's just a smooth hair kind of dog. So this is my smooth. So then what's my information? A homozygous wire hair male. So now I know that it's a homozygous wire hair, and wire hair is dominant. So I'm going to have two uppercase letters of W. I'm going to put that at the top. I'm going to put my male at the top just because I started off with a male, and it's made it to a female with smooth hair. So then smooth hair, it doesn't say if it's heterozygous or homozygous, but because smooth hair is recessive, this is my recessive one. The only way to show recessive is if it's homozygous. So it's homozygous recessive, so it must be two lowercase w's. So then I follow and form this grid, and once you have this grid, I'm just going to now fill in the grid with all these possibilities. I have an uppercase, lowercase, uppercase, this is from here, and then lowercase. And then uppercase, you always put your uppercase first, lowercase, uppercase, and lowercase. Oops. So that's my Punnett square that I've completed. Now I need to show two things about the genotypes and phenotypes. So let me just kind of change color for this one. So your genotypes, this is all about the letters, all about the genes. So if you look at these four boxes, these are all the combinations that you can have. The only combination that you can have is uppercase and lowercase. So then you have a heterozygous, and what's my percentage? Well, out of the four boxes, I have all four. So that means it is 100% uppercase and lowercase w, or 1 ratio, 1 to 1, or 100% of uppercase and lowercase. My phenotype here is, here's my phenotype. That's my physical features, things I can see. So if you look at here, I have this one that is wire hair. This one is also wire hair. This one's wire hair. And this one is also wire hair. That means it's 100% going to be wire hair texture. So 100% of this will show. All the dogs will give you that So that's the first question. Oh, and this says F1 generation. F means filial. That means your generation, the first generation. This is your family first generation of the offspring. The next question, let's read this one together. I'll give you, you can pause it and give it a try. Short hair L is dominant over long hair in rabbits. So I know that L is short and lowercase l is for long. If you want to pause it here and read it and try it out, give it a go. Next bit of information, 2 heterozygous rabbits are mated. So I know that my male is going to be heterozygous with uppercase, lowercase. My female is also uppercase and lowercase. So let's create the Punnett square. Put the male here just because I started with it, and then same for the female on the side. Form my grid of 4 boxes because these show all the combinations that I can have with my genes. Uppercase, 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 lowercase, uppercase, lowercase, lowercase, lowercase. Now you can see that this one is a bit different than the previous one. So let's start off with my genotypes. So what's my genotype? So my genotype is looking at all the letters. LL, uppercase, lowercase, and lowercase, lowercase. If we were to look at the ratios, how many out of the 4 do we have? Well, 1 out of 4 will be uppercase, uppercase, or homozygous dominant. So I'm going to put 1. And then for this one, I have 2 out of the 4. I'm going to put 2 here. And then 1 over here. So the ratio is actually just 1, 2, 1. So then you can see that's the ratio of my genotypes. My phenotypes, however, doesn't necessarily have to be exactly the same as my genotypes. Because if you look at these 3 boxes, uppercase, uppercase, that's going to be my short hair. I have an uppercase, that means it's dominant, that's also short. This is also short. So 3 out of the 4 boxes are going to be my short hair, and then 1 of them is going to be my long hair. So that's my ratio. And if I wanted to convert this to a percentage, I'm just taking 3 divided by 4 multiplied by 100, which is 75% of my offspring is going to be short, and then 25% of that is going to be my long. So these are your 2 sort of basic introductory questions to Punnett Squares setup. In the next couple of videos, we're going to show you some more advanced problems where you're not given the genotypes and you have to use some test crossing to decide, or even to use test cross to decide your probability of different genotypes and other Punnett Square type of questions. So hopefully this video has helped you. See you later.